All right, so our next video is the fluid mosaic model of the plasma membrane. So remember the plasma membrane. Okay, so we have a little cell here, right? And it's got this little gate on the outside. And that's really important, right? To have protection from everything else floating around the cell, okay? So this is what we're taking a look at. And there's a couple reasons why we call it the fluid mosaic. Fluid because it's not a static gate, meaning it's not rigid. It's not just this spot is always here and this spot is always here. It's constantly moving around. The components are moving around. And then we call it mosaic because you think of like a mosaic piece of art. It's a bunch of different pieces put together, and that's what it is. So it's not just one component, it's many components put together, okay? So let's make a drawing of what it starts as. So probably the most telltale thing when you see it is the phospholipid bilayer, right? So it's got a fatty acid tail. So these are the tails. It's a fatty acid tail and a polar head, okay? And so it's a phospholipid bilayer. Bi means two, and that's what you see here, 1, 2, okay? So it goes in this kind of orientation, and this is the gate that's starting to form around the cell, okay? So the sides that face in, they are what we call hydrophobic. Hydro meaning water, phobic meaning scared of or fearing, so water fearing, okay? The polar tails, the polar heads are hydrophilic. Philic or water loving, okay? So this is water hating, water loving. And this gives us a really good setup to be a protective layer. So we only allow certain things to come inside depending on if they're hydrophobic or hydrophilic or they're polar components, okay? And so this forms the phospholipid bilayer. So that's one of the major components. And this is what really adds to the fluidity because these guys are constantly rearranging themselves. They're moving past each other like this constantly. It happens very, very quickly, probably 10 million times per second. It's insane. Okay, then we get to the mosaic part, okay? So the mosaic part means we have a lot of different components. So not only do we have the phospholipid bilayer, so we'll label that as component 1, we also have proteins embedded in the membrane, okay? So we can have big proteins in here. And I'll mark that as 2. So we can have big proteins. The proteins can be integral proteins or transmembrane proteins. So integral in the membrane. They can be transmembrane when they go through both sides. So trans or spanning the whole membrane. They could also just be peripheral. So we could have ones that just sit on one side or just sit on the other or on the periphery. Those could be peripheral proteins, okay? So that's the second component. I'm going to draw a little bit more out here. Okay, what else do we find? So we find phospholipid bilayer, proteins. Something else we can also find is cholesterol molecules. And I'll kind of draw them as they're little molecules that get stuck in between the phospholipid bilayer. Oops. Remember, polar head groups always face out, tails always face each other. The tails like each other and nothing else, okay? So we're still drawing this outside layer right here. Now we've stuck in some proteins. Now I've stuck in a little cholesterol layer, right? And so it's starting to kind of look like this as it wraps around the cell membrane, right? So we're just showing it in a line here, but this is what it's actually doing. And so something I should mention, the inside is the cytoplasm of the cell, and the outside is the ECF, or the extracellular fluid. So this side could be the ECF, and this side could be the cytoplasm. And so this is showing you inside the cell versus outside. But do you see how it would wrap around like this? Okay, so perfect. So back to this guy. This is cholesterol. All right, so cholesterol will be our third component. And what does cholesterol do? Cholesterol is pretty cool because it basically stabilizes the membrane. So when it gets really warm, think about like butter. So butter would be like fat, right? So butter, think when it's warm versus cold. When it's warm, it starts to melt and kind of get really loosey-goosey, right? But when it's cold, or when it's sitting out on the, right, when it's sitting in the fridge, it's really rigid, right? So you have like melty butter versus your stick of butter that's very rigid. Well, your membrane works exactly the same way. So when it's warm, it gets kind of loosey-goosey, and when it's cold, these little tails stick straight up. Okay, but how do we kind of keep the membrane from kind of falling apart? Well, we can stick things in called cholesterol, and cholesterol can serve as fluidity buffers. And basically they stick in between, so think if these two guys on the side fell over, at least the cholesterol would be in the middle to kind of build a frame or a support system. So the cholesterol is the fluidity buffer with temperature changes. Okay, so when temperature changes, cholesterol is able to help the membrane out. Okay, so those would be some really important components of the cell membrane. So that was phospholipid bilayer, proteins, they could be transmembrane, integral proteins, they could be peripheral proteins, cholesterol to help maintain the structure at different temps, and then let's draw a little bit further out. What else we could find is we could find attachments here. And let's draw them in black because we're running out of colors. So we could draw carbohydrate chains. These would be like, you could think of these as being like sugar chains. And when we add carbohydrate chains to, say we had a protein here, I'll just draw them looking like little extensions coming off. This is the carbohydrate chain attached to a protein. So that, remember sugar, so that's like a glycoprotein, okay? Or the other option for the carbohydrate chain would be to attach it to a lipid, which then it would be called a glycolipid, okay? So those are some other things that we could attach to the membrane. What are they for? Well, they'll be really good as serving as sort of like name tags. So do you see how they kind of stick off the end? Well, we need to be able to identify what type of cell we are or what type of receptor we have. So what we can do is we can attach little name tags that stick out. We can attach carbohydrate chain, like lipids or glycoproteins. So this would be like the name tag that we're wearing, okay? And this would be component 4, okay? So let's put that all together. So 1, phospholipid bilayer. And the really important part of the phospholipid bilayer is it has a hydrophobic inner portion and a hydrophilic outer portion, and that's going to only allow certain things to go through the membrane, right? So things that don't quite jive with this middle portion can't get through, they're going to need help, okay? Cue the membrane proteins, okay? So the proteins can help with the transport, okay? So proteins can be really good at transporting substances through. They can act as receptors. They can act as enzymes. They can help link adjacent cells together. So say you had another cell out here, this protein could help you link together. Okay, so there's lots of functions of proteins. I'll put a smiley face here because they're my favorite thing. All right, 3, cholesterol is good for temperatures, right? So a fluidity buffer. So think of butter, warm versus cold, right? Cholesterol adds that rigid structure for shape. Okay, and then the last thing is the carbohydrate chains for glycolipids, proteins. And these serve for, they can serve as identification name tags.